Hey guys, welcome to video number 2. Like we said in the last one, in this one we're going to go through a few practice problems that look at some of the content that we taught in the lesson in the previous video. And then we're going to go through a virtual fly lab to get you some on-hand experience and really kind of figure out why what you're doing is important. So let's get started off with the practice problems. Alright, so practice problem number 1. In watermelon, solid green rim color, which is denoted by a big G, is dominant to stripes, denoted by a little g. So let's just make a little note of that right off the bat, up top here, because it's important to kind of keep track, otherwise you might get confused with yourself later. So we have big G is going to be solid, and then our little g is going to be stripes. Alright, so the farmer crosses 2 watermelon plants that are heterozygous for rim color. So remember, heterozygous means they have one of the dominant alleles and one of the little alleles. And typically a cross is written where you show the genotype of one parent and X as in cross, the genotype of the other parent. So here's our cross right here. And they want to know what the odds are that the offspring will have solid green rims. So what should we be looking for here? If they have solid, because solid is the dominant phenotype, then they should either have a genotype with 2 big G's or a heterozygous phenotype with a big G and a little g. So let's go ahead and do the cross here. So again, we have one parent with a big G and a little g, and our next parent with a big G and a little g. Here we'll draw our Punnett square. Not the best square, but it doesn't really matter. So for here we have a big G and a big G, little g, or the other way around, big G, little g here, and then little g, little g. So again, now we're looking at which ones have solid green. So solid green either has the 2 big G's or one big G. So we have one right here, one right here, and one right here, but not this one. So we have 3 out of 4. And the answer is going to be D. So that's a simple monohybrid cross that we're going to see pretty often. Alright, now here's a little bit more practice with a monohybrid cross. So many farmers prefer cattle without horns because it is safer for their herds. The allele for no horns, capital N, so again we'll write that. We'll just put no. And then is dominant to the allele for the presence of horns. So then little n is horns. So now they're looking for the chance that the offspring will have horns. So we're looking for the amount of offspring that have n, 2 little n's, 2 recessive alleles. And what are we crossing here? We are crossing a male with horns, so therefore they have to have an N with a heterozygous female without horns. So they have big N, little n. All right, let's go ahead and do it again. So we have little n, big N, little n. And again, obviously, it doesn't really matter which side you put them on as long as you include both and they're the proper genotype. So now we have a little n and a big N. And it's just common notation to always include the capital letter or the dominant allele first. And we have another big N and a little n. Little n, and little n, little n. So now here we have 2 of the little n's. So we have 2 out of 4, or 1/2 if we reduce that, giving us our answer, A. Alright, so now this one's going to be a little bit more complicated because, as you can see, we're looking at 2 different traits here. So let's just make sure we keep track of everything, and we should be alright. So we'll do the same thing as usual. So in rabbits, white fur, capital W is white, is dominant to black. So then little w is black. And long ears, capital E, we'll just put long, are dominant to short ears, little e, and we'll just put short for that. So a breeder mates two rabbits that are heterozygous, and it shows the genotype there for both traits. So we're going to have two heterozygous parents crossing. All right, and it is asking, what is the chance that the offspring will be black with long ears? So what kind of genotype would be black with long ears? Black is recessive here, right? So in order to have a black phenotype, we would need to have two of the little w, two of the recessive alleles. So we're going to have w, little w, and then it's asking for with long ears, which is the dominant phenotype here. So now here, we can either have two homozygous genotype, or we can have a heterozygous phenotype. And both of these would be black with long ears. So let's do the cross. And this is going to be the first time that you see a dihybrid cross in a Punnett square, so make sure you give yourself enough space as it gets a little bit complex. So what we're going to do for this one, obviously because we have two traits, we have to make it a little bit bigger, is for your, here we'll start off, try to make this as clean as possible. So you're going to have 4 rows and 4 columns. And basically, each of your columns and each of your rows is going to contain one allele from each trait. So for example, or for our top one, we're going to have this big W with this E. We're going to have the big W with the little e. We're going to have this with this, and then that with that. So it's going to look like this. And then, so you see what I did there? So this one has our two dominant alleles, this one and this one. Next one has the first one and the last. This one has this one and this one. And then this one has this one and this one. And now because they both have the exact same genotype, we can do the same thing along this side as well. Otherwise, you would have to go through the same process and make sure that you're writing it down properly. My pen is a little bit delayed here. All right, so now we're going to do the same thing as usual. We're just going to put the alleles together here and try to keep the traits together. So keep the W's with the W's and E's with the E's. And remember, we're looking for either of these genotypes. All right, so now we have finished our Punnett square here. I know it's a little bit unclear, but sorry, my pen was lagging a little bit as we were doing that, so hopefully you can still get the gist of it. So little e's might look like little c's here, but anyways, all the same. So again, what we're looking for is they're all going to have 2 of the small w's because we're looking for black, and then they're either going to have homozygous dominant alleles here, so the 2 long ear alleles, or 1 long ear allele and 1 short ear allele. All right, so the top row and the top column, they all have at least 1 dominant fur allele. So none of those are going to be right. And then same thing right here. They all have at least 1 big W. But now here we have 2 little w's and 2 big e's. That fits our requirements here, so we'll circle that one. And then we have, again, this one fits our requirements. And then this one as well fits our requirements. Does this one? It does not. So we have the 2 little e's, which would give it short ears, and we're looking for long ears. So now we have 3 out of 16. So when you're doing a dihybrid cross, there's always going to be 16. Wow, this pen is really lagging. All right, so 3 out of 16. That should be our answer. Letter D over here. So we just went through a few practice problems here, obviously, but you can do lots of practice at home. There's tons of examples all over the web. All you have to Google is practice problems for monohybrid and dihybrid crosses. So I definitely encourage you to do that. And aside from that, we'll get right into the virtual lab. So for the introduction, in this laboratory you will study the patterns by which physical characteristics are transmitted from generation to generation by breeding fruit flies. So, and here is the actual name for that, Drosophila melanogaster, of unknown genetic composition and studying the traits and ratios seen in their offspring. It will determine whether a trait follows a monohybrid or dihybrid pattern of inheritance and whether it is sex-linked or autosomal. You will use statistical analysis to support your conclusions. And the statistical analysis is going to be the chi-squared that we learned at the very end of the lesson. All right, so some key concepts before we really get into it here. We say that like comes from like, but offspring do not always look exactly like their parents. In this laboratory, you may breed two parent flies with red eyes and get some offspring with sepia, deep brown eyes. How is this possible? We already looked a little bit into this stuff in the lesson, so some of this might be redundant, but just for the sake of understanding the lab itself, I'm still going to read through it for you. In the 19th century, Gregor Mendel discovered the principles of inheritance through a series of careful experiments with peas. We know now that physical characteristics are transmitted from generation to generation according to some general patterns. When we have sufficient information about the parents, we can predict the occurrence of traits in offspring. By analyzing the offspring, we can discern the mode of transmission, monohybrid or dihybrid, sex-linked or autosomal. And then down here we just have the necessary knowledge that you're going to need to be able to complete this lab. And as you can see, we already went through this, we went through that, we went through that. The only thing that we didn't really talk about was the crossing over and linked genes, but don't worry, you shouldn't need to use it for this lab. All right, so designing the experiment. The first thing it's talking about is how to differentiate between the male and the female flies. So the real main difference here is going to be that the males are like shorter, a little bit shorter but wider, as you can see here. And then the males also have this comb on their leg that the females do not. So those are the two tells that you're going to be able to have. This shouldn't be too necessary because it should make it pretty clear, but that's just some information that's going to help you with this lab. So now it kind of tests us a little bit just to make sure that we're capable of going on. So is male or is fly A a male or female? We see the comb, it's going to be a male. Is fly B a male or female? Longer body, no comb, female. Same thing here, longer body, no comb, female. Comb, shorter body, male. And then comb, shorter body, male. So we should get nice all correct there. All right, good stuff. We'll move on. And now this is going to be the procedure of the lab. Following directions in your lab manual or your instructor's directions, over a period of 4 weeks, you will study 3 generations of flies. Now keep in mind, this is all virtual. We're not actually doing this in class. You begin with a parental pea generation that has already mated. The figure below gives you an overview of the procedures you will follow. You may want to print it so you can refer to it through the rest of the lab. You won't need to do this. If anything, you can just easily backtrack and take a look at it later, but you can print it if you'd really like. Once you've studied the overview figure, continue to see more details of the different stages of the experiment on the succeeding pages. So here we're starting with our pea generation here, and this is going to be our homozygous parents, right? We breed those together into the F1 generation where we're going to get the heterozygous F1 generation. And then we remove the parents from there, breed the F1 generation, all the heterozygous flies together, and then we get the F2 generation. And then the F2 generation is the one that we're going to be examining the ratios in really specifically. So this is just listing some common mutations that we're going to see in this lab. And really there's just 2 traits here. So we have ebony body versus wingless. So 2 different alleles for 1 trait, and then white eyes versus sepia eyes, and that's just another 2 different alleles for 1 trait. And we'll see in a bit whether one's dominant or recessive. And a little bit of an activity before we get into the real juice of it. So it's asking why it is important to remove the adults in once we breed the F1 generation. So as you can imagine, our P generation, the parents, they actually have purely homozygous traits, right? Whereas our F1 generation is going to have the heterozygous traits. So we don't want our homozygous P generation breeding with our F1 generation there, right? Because that's going to skew our results. So we want to remove them so we have a pure F1, purely heterozygous generation. So type that in there. We'll check our answer. And it says it is important to keep generations separate so that you know you are crossing only F1 flies. Basically what we were just talking about. Now, for the next section of the lab, the flies that hatch from the initial vial are the F1 generation. Immobilize and carefully observe these flies, note and record any genetic variations. So this is just explaining what you would do in the real lab. Now it's asking, what generation will the offspring of our F1 generation be? And this is something that we talked about in our notes. So remember, we go from the P generation, the parents, to the F1 generation, and then once we breed the F1 generation, we get the F2 generation. And there we go. All right. So in this section, one way to discover patterns of inheritance is by working backward. In other words, you determine the genotype of the original parental generation by careful analysis of the F1 and F2 generations. Now remember, this is actually something we talked about in the first video, where the purpose of doing these monohybrid and dihybrid crosses is to figure out the genotype of the parental generation. So let's examine two sample cases that trace eye color. For each, look at the data chart with the number of male and female flies exhibiting each eye color, then answer the questions. So here it gives us a nice table of results for our F1 generation. You can print them as well, or you can copy and paste them, screenshot them. They're not super necessary, but it might help you if you really want to keep up with it. So it's asking us here, based on the data obtained, is this cross a monohybrid or dihybrid cross? And if you can see, we're really just testing one trait here, right? One trait that has two alleles. One allele is the red eye, and the other allele is the sepia eyes. So because we're only considering one trait here, it's going to be a monohybrid cross. Now, is the cross sex-linked or autosomal? So really here we should be looking for a surface feature. Are there any significant differences between the females and the males? Now notice, yeah, there are a decent amount more males that are red eye than sepia eyes. But there's no real indication here. They're still relatively close. So without more information, we can pretty well say that it's going to be autosomal. We'll check our answers. And we're correct. So this one is fairly obvious. But for this one, since the pattern seems to be the same for males and females, the inheritance is probably autosomal. And remember, autosomal is just not sex-linked. All right. And now for this one gets a little bit more complicated. So based on the data obtained, which is the most likely mode of inheritance in case 2? So let's take a look here. We're looking at autosomal or sex-linked again. So in our F1 generation, we have relatively the same number of males and females, and they're all red eyes. Whereas in the F2 generation, we have no females that are white eyes. So I'm going to take a second and write this out for you. The first question was asking us if it is autosomal or sex-linked, the cross. So let's take a look at this here. If the cross were autosomal, odds are we would probably have a relatively even distribution between males and females in the F2 generation. And as we can see right here, we have 21 females with red eyes, and then for males, we have 12 with red eyes and 8 with white eyes. So that's already kind of pushing us away from this autosomal assumption. But now let's consider sex-linked. So here we have no females, right? So that means maybe it's possible that the female was a carrier in this generation for the white eyes, and then gave those alleles to the male. So let's check this out here. Let's assign a big R to red and a little r to white. And first we'll just answer the first question here. So this is going to be, we can pretty confidently say that this one's going to be sex-linked. As the odds of this happening, if it was autosomal, are very low. Now the parental genotypes. So let's go with the strategy that we were thinking of here. So let's say in this generation, our female, they have red eyes, and red eyes are probably dominant. So they're at least going to have at least one red allele. And then females have two X chromosomes, right? So then let's say they're a carrier for the white allele here. And this is the notation we use when we're doing sex-linked crosses, because it's important to make sure that you're keeping track of the males and the females in terms of X and Y chromosomes. So anyways, so let's say in our F1 generation, our female was hypothetically a heterozygous for the red and the white trait. Now they're being crossed against a male who has red eyes, and a male only has one X chromosome, right? So this is automatically going to be a big R. Now let's go through the Punnett square for this. So we have the male, and we have the female. And again, my pen is lagging a little bit, I apologize. So for here, we're going to have two X's, a female with two big R's, and then we're going to have an X with a big R and a Y, so that's a male with red eyes. Here we're going to have an X with a big R and an X with a little r. So a female that's a carrier for white eyes, but it still phenotypically has red eyes. And then our last one is going to be a male with the Y chromosome with a small r, so they're going to have the white eyes. So as we can see here, it's impossible for a female in this generation to have white eyes because no matter what, they'll only have one white eye allele at most, and white eye is recessive. Whereas for males, 50% of them, which is pretty accurate here because we have 12 and 8, are going to be white eyes. So the parental genotypes are then consistent with our first hypothesis, just like that. All right, so from what I remember, I believe they actually had this one wrong here. So I'm going to put the right answer down anyway. And as we can see, yeah, so this is the lab, actually, that did it wrong. And it's of course, it's saying to make a Punnett square, which we kind of just did. So ignore that. It's just a mistake in the actual lab itself. But this is, as you saw, this would be the right answer there. So now we're going to look into a chi-square analysis of the data. So if you kept the data, the tables from case 1, now would be your time to bring that up. And if not, you can always just refer back to it in a little bit. So we're going to do a little bit of practice now calculating the chi-square for this experiment. And this is just a review of what we talked about in our lesson. But I'll read it out for you just to kind of get another little bit of an application for it. So in a cross between two heterozygous individuals, the offspring would be expected to show a 3:1 ratio, right? Because this is a monohybrid cross. We're going to have one individual with the double homozygous dominant trait, two heterozygous individuals, and then one double recessive, so homozygous for the recessive trait. For example, in case 1, 3/4 of the individuals would have red wild-type eyes and 1/4 would have sepia eyes. If there are 44 offspring, how many are expected to have red eyes? So there is going to be 3/4 of the individuals that have red eyes because this trait is dominant. So we can do 3/4 of 44, we get 33. And then same thing for the sepia eyes, we just do 1/4 of 44. And now we have our expected values and we have our observed values from the table before. And now calculating the chi-square, exact same formula that we showed in the lesson before. And if you have this table, if it works on your computer, for some reason it doesn't work on mine, I urge you to fill it out, but we'll skip it for now because it doesn't seem to be working. All right, and now again, this is reviewing some of the stuff that we already looked at. Degrees of freedom, we already know it's one less than the total number of classes or outcomes. And then the p-value here as well. And I'll read through their definition just so you can maybe get a little bit of a better picture. So the probability value is the probability that the deviation as great as or greater than each chi-square value would occur simply by chance. Many biologists agree that deviations having a chance probability greater than 5% are not statistically significant. Therefore, when you calculate chi-square, you should consult the table for the p-value in the 0.05 row. And here's an example of the table that you would use. So you don't need to construct this yourself. This will always be given to you when you're solving a problem like this. Or you can easily look one up. And now it is asking us to determine the degrees of freedom. This is the number of categories, red eyes or sepia eyes, minus 1. So remember, we're looking at case 1 here, and it's a monohybrid cross. So how many potential outcomes there are, and we're considering the phenotypes, right? So it's either they have sepia eyes or they have red eyes. So there's 2. That means that we're only going to have 1 degree of freedom because it's the amount of classes minus 1. And now it's asking us to find the probability value for 1 degree of freedom in the 0.05 row. So we have 1 degree of freedom, and it's asking us for the 0.05 row, because this is where we really want to hit to be statistically significant. And the value here that we really want to be under to get 0.05 is 3.84. Now, usually when you're doing this and you calculate your chi-square value, you're usually not going to have a value that's exactly any of these. So you're really going to be in between one of the percentages probably. So we'll check our answers and we should be good. Perfect. There we go. All right, and now here is the lab quiz. So these questions we're going to do mainly on a PowerPoint, so I can draw it out and we can really figure it out together here. But for now, we will try to get through a few together. So on the basis of the results shown in the table, which statement is most likely true? Our options here, the genes for red eyes and normal wings are linked. This is a topic that we didn't go through, so we can just X that out to begin with, because I know that it won't be this case. The gene for no wings is sex-linked, so let's look at this option here. So if the gene for no wings was sex-linked, we should see a significant difference between the males and females in the offspring for it. And as we can see, the ones with no wings, pretty similar there. And again, not totally different there. And there would be a huge diverse outcome if there actually was a sex-linked inheritance there. Then the gene for red eyes and the gene for no wings are both dominant. So if the no wings was dominant as well, we'd see a lot more of these red eyes and no wings. But instead, we see a lot of these red eyes and normal wings. So odds are that the red eyes and normal wings are the 2 traits that are dominant in this case. Next option, the gene for eye color is inherited independently of the gene for wings. So remember, this was Mendel's law of inheritance. And there doesn't seem to be a linkage between any 2 of these 2 traits. And we didn't really discuss that. So we can rule, so this one is looking pretty accurate right now. And then the F1 mates were both homozygous for both eye color and wings. So if they were both homozygous, we'd probably have only heterozygous offspring. So in this case, that can't be right either. So we're going to go with answer D here. Now, number 2, if the parents were both heterozygous for eye color and wings, what phenotypic ratio would you predict for the offspring if these traits are not linked? So, if we remember, we should be seeing our typical 9:3:1 ratio if there are 2 traits with 2 alleles and one is dominant and one is recessive. And we're breeding 2 heterozygous parents here, right? So that's our typical dihybrid cross inheritance pattern. All right, so now for the next 3 questions here, we're going to do them all on the slide just so I can walk you through them a little bit. And they're all connected here, so we'll just do them in a nice flow. So open up this PowerPoint. All right, so the first question, well, question number 3 was to calculate the chi-square value. And we're going to be doing it for this table here, which is basically what we're trying to do here is we're trying to see whether or not that this inheritance pattern is due to a regular dihybrid cross or if there's another phenomenon happening. So as you remember from the lesson, our chi-square formula is going to be the sum of all of these. And this is observed minus expected squared over expected. And so the first thing that we can do before we make our table over here is that we can count up the total number of offspring here. And here we're just going to add up all the males and all the females because the inheritance shouldn't be different for each of them. So I already added this up in advance. So in total, when you add up all of these numbers, you get 160, which is a really nice number to work with in this case. And remember, we're looking for that typical 9, 3, 3, 1 ratio. All right, so let's start making our table here. So we'll make a column for each of our phenotypes here, and then we'll add them up here. So our first column, we'll do red eyes and normal wings, and we'll just make it look like that for now. It doesn't really matter that much how you denote it, just as long as you pay attention. And then we'll do red eyes and no wings. We'll do the sepia eyes and normal wings, and then the sepia eyes and no wings. All right, so and then in our table, we already have our observed values. So our observed values for each of them are just going to be the number for the males plus the number for the females. And same thing for the rest of them, 13 plus 9, such. So what we do need to calculate, however, is our expected. And then after expected, we'll just do it in steps to make it easier for us, and then we'll calculate our observed minus expected, and then we'll do our observed minus expected over expected. Sorry, my pen is lagging yet again a little bit, but hopefully you can make out what's happening there. So our expected value is going to be 9 out of 16, right? Because it's a 9, 3, 3, 1 ratio. Together these add up to 16. So then the red eyes and normal wings should account for 9 out of 16 of the total amount of offspring that they are. And in our sample, we have 160 offspring. So that's why I said the math is a little bit easy, because 9 out of 16 translates to a nice 90. And then same thing for the other ones. For the 2 3s, they're going to be 30 because it's 3 out of 16. 30 and 30. And then the last one is going to be 10. All right, and now for these, we're basically just going to do simple subtraction. So again, your observed value is going to be, in this case, 50 plus 48, so 98, and then 98 minus 90. So we're going to get 8 here. Then this one's going to be minus 8. This one's minus 4. And then this one is 4. And then again here, just a matter of plugging into the equation. So just square the value that you received in this row here, and then divide it by the expected value. And just use a calculator for this, because you probably won't be getting a very clean number. So for this one, we have 0.71. So we have 2.13. And again, I pre-calculated these, so I wouldn't have to waste time in the video, which is why I'm going fast, but feel free to pause and take your time. And then 1.6. So then again, to get the chi-square value, we have to take the sum of all of these. So 0.71 plus 2.13 plus 0.53 plus 1.6. And when we sum those, we get 4.97. That doesn't look very great, but 4.97 is the answer to this one. And that should be one of the answers on the online portal. So now we'll move on to the next questions here. So degrees of freedom. Now remember, we're looking at the way we determine degrees of freedom is basically by the number of expected outcomes. So basically the phenotypes minus 1. So in this case, we had the 4 different phenotypes, right? We had the red eyes and normal wings, we had the red eyes and no wings, we had the sepia eyes and normal wings, and the sepia eyes and no wings. So that's 4 in total, and then minus 1, we get 3 degrees of freedom. All right, and now, so we have our 3 degrees of freedom, and then we have our chi-squared value, that is 4.97. And now we can go look in the table here. So go under the column that, again, we're looking for it to be significant, we should be looking in this 0.05 column. And this is just a standard value to determine if a result is significant. So again, we have 3 degrees of freedom. So we're going to be looking at this column right here, and then we're going to be trying to get this 0.05. So for us to say our results are significant, we should have a value, our chi-squared value should be less than 7.2. So all these values in the table here are the chi-square values. And in order to have a 0.05 under the 3rd degree of freedom, we need to have our value less than 7.82. And as you can see, we had 4.97, less than that. So we can say that our p-value is less than 0.05, making our results significant. So now we'll go back into the online portal here. So our answer for this one, 4.977. Our degrees of freedom, 3. And for this question, so A says, since the calculated value for chi-square is less than 7.2, the results support the hypothesis that the parents are heterozygous for 2 unlinked traits. And that's exactly what we just found at the end there. So we'll select that as well. Check our answers. And sure enough, we are all good. All right, so hopefully that gave you some good practical experience. Our next video is going to be on how to write a lab report. So in doing so, after doing this virtual lab online, you're going to be putting those results into some tables and making a formal lab report. So like I said, this next video is just going to kind of walk you through all the different sections that you need, what you're supposed to include, and what is expected of a good formal lab report. So we will see you in the next video.